Hello, this is Stephen Friend. I am very, very sorry to not be joining you in person. For family reasons, I'm not able to be there. I'm on the west coast of the United States, and I'm going to talk to you about some examples of opportunities or ways to exit from silent despair. Hope that fits with the topics that you're doing there in Amsterdam. So we're surrounded by aspects of despair, whether it's thinking about the climate or whether it is thinking about what's going on in the Middle East or things that are close to us in terms of what's happening in our communities. What I want to do is give you two tales. I want to give you two tales of tools or approaches that seem to allow ones to exit despair. I'm going to talk to you about one on detection and one about intervention. So let's talk about on redesigning detection. Um, when anyone goes into a clinician's office, they are expecting that clinician to, in a moment's time, be able to see where you are and get a sense of what your symptoms are to diagnose you in a really static way that's constrained by our current definitions of symptoms and also sort of like our expectations of what are there. And for that reason, now almost 15 years ago, we began to ask, let's use our technology. Let's ask whether some of our devices could do what a clinician does, which is to use their touch and their hearing, their senses, sight, taste, and smell. And could it be possible to actually collect that information without a clinician in the loop? It would be certainly more affordable. It would be one that could be used in many different areas in different ways and done continuously. So along those lines, we began to ask, could you do semi-continuous sensing from wearables that would collect physiologic signals, whether it's from a ring or an app or a phone, and back then, 2012, 2011, 2012, we did some simple tests to show that an app on a phone could measure various aspects of Parkinson's disease very well. And in fact, as you did that, you could see there was no average human, no single measures. And depending upon what you asked that person to do, you could get different measures of their disease. So, in a broad sense, yes, this was possible. And by 2015, we began to build tools that are now widely used that allowed researchers to work with patients that weren't at all close to them at all. Anywhere in the world, those individuals could be being connected with a device and being part of studies, this research kits strategy. And with that, I was lucky enough to go into Apple to work with Jeff Williams and others on strategy and had a remarkable time sort of understanding more about what wearables could do. But by 2017, I saw that much of the world was headed towards extending the doctor's office, not really in giving agency to individuals. And what we were interested in doing was asking, could we build a personal health assistant? Could we figure out a way to self-navigate before and after symptoms arise, nurturing actions that were helping in times of strength, and do it by bringing in and asking individuals to do it by each other for each other. So formed a nonprofit called For You and Me, got connected with Oxford University. And what I want to tell you about is this work that we've been doing at About For You and Me. So it's a nonprofit organization. We're asking how can we use smartphone apps and wearable devices as a way to empower individuals, to give agency back to individuals to self-navigate, to work on ways that participant-driven research and co-constructing health navigation tools could occur. And for all of this, we would put that data in the public domain along with the IP, et cetera. So what have we been doing? We've been trying to shift from signals to symptoms, where we would use various aspects, tools, sensors on the various devices, and use them to weave together a way of looking at symptoms like cognition, mood, sleep, gait, and fatigue. And we've been doing a fair amount of that. We also said, let's blow up the construct where from medieval ages, people in studies were subjects, and let's see if we can make it so that there's a symmetry between the roles of the study participant and the researcher. Can we put them on equal footing and work in listening to study participants in their anecdotes and be able to capture it in a quote participant-centered research study? So we've done this more than 10 studies that have been completed. There's a bunch of papers that have come out. And it's led to a chance to do some fun things now. We're working with the Chan Zuckerberg Initiative to work with rare genetic diseases, called Rare is One, that obviously is not to one, but rare genetic disease advocate groups. We're trying to see if we can empower patients and their families to have better ways to follow their symptoms and navigate their disease. Secondly, to see if there are ways that patients' voices can better come out when they're talking to their clinicians. And also to evolve to where the patients and their advocates could actually be designing the apps themselves and them choosing what they want to do, why they want to do it, etc. So for this co-design evolution, we started, as many people do, going like, oh, how could this be helpful to a researcher? That's important. And then we realized actually this doctor-patient dialogue was going to be pretty important. But then what began to happen, and it was really the patient's voices, was we realized that being able to do something that was for the patient because the patient wanted to know it was good, and not just retrospectively, but what we're now calling, can you build a compass which allows someone to say, if I do this, this will make me better or make me worse. And so to do that, this co-design with these groups has been fabulous, where we work together and then we come up with an app design and they try it and they see if this will work or not. And so we've been adding in layers of data, not just the passive data in the surveys, but times when someone could stop and collect a momentary insight, a way where when something is happening and the person wants to be reminded if something goes astray, a little nudge could come to them. And then very importantly, looking at evening reflections about where is this going? Is this working for me or not? That is one of self-reflection. Just as an example, imagine that if you had a way by video or audio or text or drawings to follow what it is that's happening to you and then be able to bring that up on your large computer screen and match that with what was going on. That's the type of things that we've been trying to do. And the patient advocates have been saying, like, can we do it in different ways? Like, can we do journaling? Can we do coloring pages? And it's really, I think, given a rich way of collecting that data. At the same time, we have been looking at wearables in cancer for those who are interested. We did some hero studies about helping enable real-time observations with pancreatic and high-grade gliomas. And our major goal was to see when could we get to where, because someone has some tumor burden, the way they are behaving, the way they're carrying out their activities might be a clue that actually something was going on that would then allow them to get a blood test that otherwise might not be available, and also to track the effects of chemotherapy. So I've gone through this quote detection story. I hope you've noticed that the power of the active patient roles, by being present at the helm of the ship, not just to comment on it, are what we note just quite efficiently allow them to exit their silent despair. So it's the active patient roles that are allowing them to do that. Second story is one on redesigning intervention. Same concept, but in this instance, it's looking at the absurdity of the costs of drug discovery. So I've been doing various projects for a while, over a half century, and much of that time has been trying to understand disease or work on drug discovery. And I have been rather frustrated at much of that. The example I want to give is the specific one is when at Children's, as a resident, I had a chance to see a patient that had retinoblastoma, and their father had a tumor in the same eye like them. And that grief was one that helped me motivate to going like, how could we find how that happens? Could we identify the gene that was responsible? So this is 1986. But here's the point I want to go to. 4 decades later, whether it's for retinoblastoma or Duchenne's dystrophy or thousands of other rare genetic diseases, there are no available therapies. And this is horrible. And the work that we've been doing is to ask, are there new ways to sort of break that log jam in terms of that absurdity? Those who are close to drug discovery might smile here. Those who aren't, I hope you can see that this concept of going from an idea all the way through to registration has a lot of loops, it has a lot of steps, and the most important is a long, redundant road to new medicines. As many of you already know, that process of going from target ID through to lead discovery to developing a drug takes a super long time, costs very large amounts of money. That does say £1 billion. Over 90% of the drugs that make it into the last phase 2, phase 3 of development end up failing because they do not have the intended beneficial effect that everyone had hoped they would have. And 90% of this is done in this closed, non-sharing, proprietary ecosystem, which is really because investors are trying to make a profit off of what's going on. So what we've been considering is, what would it take to build a novel drug discovery platform from identification all the way through to delivery of drugs? Could you make one where the targets are picked that are validated to have a desired beneficial effect up front? Could you ID targets that are being able to be identified as targets by analyzing the existing genome data? Could you make it so that the early work, and in fact, as I'll show you, the late work was driven by the patient advocate groups, not by investors? Could you make it so the data and the compounds would then be made open source, available to be made by anyone to manufacture, and have no patents filed on the drug? So that's the project that we've been working on. I need to ground you quickly on genes and mutations. We moved from having no idea how genetics worked in Mendel's days to the structure of DNA to the first human genome sequenced by 2001 to where now there are 4 million human genomes that are already sequenced. At the same time, these rare monogenic alleles have been increasing to where now there are more than 4,000 of these times where there's a disease gene known, but because it's a disruption, they're not druggable. So we're trying to figure out, are there new ways to find druggable methods for undruggable targets? And the solution comes through from looking at evolution from yeast to humans. The theme here, this is the most important phrase here, is specific changes in second genes can suppress symptoms of the original gene. So if you have a normal genome, let's say you have mobility, you alter gene A, you don't have mobility. If you alter gene A and there's another altered gene, usually there's still no mobility. But sometimes when there's an altered gene A and you have no mobility, and then you have something else that is altering a particular gene A, you regain mobility. So these are what are called second site suppressors. And our job is to find what are those second site suppressors and develop drugs for it. Just to see how it should be doable, this is a nice example from the lowly yeast in which someone has built a map to show how many times disrupting a second gene can modify the function of the first if you do it at saturation mutagenesis. And so we wanted to know, could we find second site suppressors that could represent ideal targets that would have the effects of being knocked out by a novel drug? How could we find them? How could we do that? Would be to look among the healthy, not those who have disease, and could we identify what it is that was driving that change? So 10 plus years ago, we did a project called the Resilience Project, just as a proof of concept. Took 500,000 genomes, looked through it and learned a lot. But most importantly, among those, quote, healthy individuals, 13 candidates came up, which were individuals carrying expected, lethal, highly penetrant alleles, but they were not noted as having any symptoms. So something was in them that was making them resilient. So these days, sorry, we are looking to build an open platform to deliver therapies, starting with working with the rare disease advocate groups, engaging them as partners, using these digital tools that we have to scan through exomes, then using new emerging tools to sort out what could be an extragenic suppressor, and then working directly with the rare disease groups so they're partnering with the regulator to complete the phase 1/2 trials. So it takes us from the finding the individuals to finding the second site suppressors, to validating them, to making probes, small molecules and antibodies on through. We're trying to build that pipeline with a central coordinating team without having investors needing to reap a profit at the end. Can we do it at first in a way that allows that to all happen? And the goal there is to go back to all those rare genetic diseases, many conditions, and see if there is a way to bring that to the point where wherever you are in the world, there's a way to deliver that drug, not just for rare genetic diseases, but for other diseases using the same model as we evolve it. So the moral of the intervention study or story is, again, same thing. There's an extreme power of being present at the helm of the ship to steer, not just to comment on it. And this will hopefully disrupt the profit-driven current methods of drug discovery and allow us to exit our silent despair. So the message here was building ways where individuals in silent despair have a way to actually be actively part of the process, not listening, not, quote, seat at the table, but in both these examples, with the wearables and for detection and these aspects of drug discovery intervention, going, why can't we build a system that's outside the current system that is allowing full control instead of being excluded? And I hope that almost made sense. And again, sorry that I'm not there. Looking forward to hearing back from you later.